Department head of the Mathematical Sciences Division, one of the largest divisions here on campus. Tomas is one of the top researchers at the university, very successful, and he actually, I just talked to him on the phone. He's running a little bit late, but he also forgot to put a tickler on that the seminar was today. So we think he's talking about databases for dynamics, a new approach to model gene regulatory networks. We're going to find out when he arrives if that's actually the seminar he's giving. So stay tuned for that. And while we're waiting, are there any questions or issues or concerns about anything? Yeah, sure.
If you are a master's student, you like this semester, or you have a PhD with nothing involved in this semester, you need to send an email to register for the MS company. This is the last time.
Great. Other things to talk about that are coming up. So on Friday are the Stibitz and Wilson Awards. For those of you who don't know what these are, there's an organization in town called the American Computer Museum. It's run by George and Barbara Kermachev. It's actually a world-class museum. And for about the last 15 years, they bring in various pioneers in both the life sciences, but also in computation. So on Friday, one of the guests will be David Ferrucci, who is one of the, basically the driving force behind IBM's Watson. That's the computer that sort of was the Jeopardy champion several years ago. And so there will be two events that will be of interest. At 3 o'clock in the Procrastinator Theater, there will be sort of a question and answer panel. I'll try to set something out closer to the time. And then at 6:30, all 5 of the awardees, both the people getting the Computer Science Stibitz Awards, but also the people getting sort of the Wilson Life Science Awards, will all be presenting sort of a brief summary of what their accomplishments are. They'll be answering questions from the audience. And then the Wilson Awards are named after E.O. Wilson, who is a world famous scientist. He sort of made his name with sort of like studying ants, but he's since sort of progressed to a much more holistic view of how kind of life and sort of ecosystems work. He's 86 years old, but he will be there, and so he will be speaking as well. So that's really a good opportunity. Friday night at 6:30, it's in the sub ballrooms. Next Thursday, if you are looking for deployment, is the career fair. So it's 9 a.m. to 3 p.m. over in the sub ballrooms. If you are wanting help with resume critiques, I think our AWC club on Thursday night at 5 o'clock is having their sort of bi-monthly meeting. And the purpose is to talk about, hi Tomas coming in, to talk about best practices with respect to resume writing. So just out of curiosity, is this the title of your talk? Okay, I've been giving people a little bit of
I'm sorry,
context here. No,
here's the more address.
so yeah, so you can plug in there and then we're set up here. Great, and so actually, Tomas, I already introduced you about 3 minutes ago, and so as soon as you're set up and catch your breath, you can take it away here.
So yeah. How do I get to?
Oh, so let's see. So you put the drive, the thumb drive in there.
I just see a lot of computer science guy. This should be on. That's what we're looking for. It's supposed to do a PowerPoint.
From here. Yeah, great. We'll get one of these lights on. Yeah, thank you so much. So what we've learned from this is we're always sending reminders to our speakers from now on.
I'm an old guy by now. Okay, so
you could just X out with that. Okay.
Just a arrow.
Okay. All right, so I'm going to try to talk to you about work which I've been doing over the last couple of years with my postdoc, Greek colleagues, and somebody from Rutgers. Okay, so here's a set of problems which I think we're facing. So I'm doing math biology, right? So if you're looking at biological systems, regulation, but also problems in combustion neuroscience, if you want to describe these things, you need to use a lot of variables, right? Thousands, actually, maybe this is a big deal concept. What you also, in biology, what's also problematic is that you do not know the fundamental laws. Right? If you're a physicist, there's some kind of understanding of that, even if force and acceleration are related. You can write down some fundamental laws and write differential equations, something which describes the system using those laws. In biology, it's not really clear by multiple proteins or genes or regulatory network what is going to, how do I describe these things in kind of a dynamic way. Okay? Typically what you do if you want to model a biological system in time, right? And there's obviously things we want to recognize and cluster because this is static things, but if you want to model behavior in time, we do what we used to do for 200 or 300 years. You find some initial conditions, and you run your differential equation model forward for some few parameter values. You have no idea how much of the behavior of the system you captured. We'd like to understand much more broadly of what the system is capable of doing, where you do not quite know what the nonlinearities are, you don't quite know what the parameters are. These are very difficult things to measure. Okay? So what I'd like to talk to you about, and this talk is a little more mathematical perhaps, so I'll try to go through this quickly, through some of these issues quickly, to describe a methodology which allows to describe this system of genes and proteins on a level which is compatible with the modeling precision. So the first, and then I'll probably talk about it later. When biologists model the behavior of the system or expression levels, if you get data which tells you it's 10 or 100, you're lucky. That's very precise. You never get 2.3, 2.5, that's the number. So it is kind of this very rough and approximate data you get to train your model. You'd like to say something perhaps on the same level you not expect to say. You cannot get garbage in and something out properly. It's so nice to be described in very rough terms, but that's something which allows you to make some conclusions. So to describe what we have been doing in dynamical systems, this is differential equations for hundreds of years, maybe not hundreds of years, maybe since Poincaré, 1900s, is something like this. So I have a parameter space, I have a set of differential equations which are parameterized. So there's different constants which are in there. Once you fix that number, that set of numbers, this could be 10, 15, that's a 100-dimensional space. You fix those numbers you're looking for, and this can be another 100 dimensions with differential equations, you're looking for so-called invariant objects, equilibrium, periodic orbits, maybe chaotic dynamics, object like that. That tells you how to organize your knowledge about the system. So for instance, in this case, for a parameter here, I'll have a 1, 2, 3 equilibrium, and dynamics will kind of go, this is stable, this will be unstable, unstable equilibrium. That description is kind of ideal which you can get. Now, this is very non-computable item. Okay? If I have a 100-dimensional system here, 100-dimensional systems here, it's not feasible to describe that or to understand this, and probably not even desirable. Okay? So, again, I'm not going into details here of how we do this, but this is a typical network you get in biology. Usually you get these, they call them hairballs. You go and get science magazine and these really large things which are connected, right? And that's the outline, and it's the output of an article. And they go like, this is what we got. And you say, well, okay, what do you do with that? Well, how much knowledge you can get out? So this is a very small model. But there are some elements. This could be genes, proteins, regulatory elements. The arrows are of two types: the up regulation, which is a sharp arrow, and down regulation, which is a red dull arrow. Okay? And those are relatively easy to get. Right? You get the X, Y, Z things from genetic screens, high throughput methods. You get these arrows that are a little bit more difficult to get, but there are also software ways how to do this. But also, if you knock out one of these elements, and if Y2 goes up, you say, oh, that thing was a down regulator. If you take it out, it went up. If you take it out and things go down, you say it's an up regulator. You notice this is that's not quite how it works, because you never know whether there's 3 or 4 things in between, but you just know that's what they do. Okay? So you come up with this. There's a lot of methodology how you analyze these hairballs just based on a graph theoretical ideas, right? What's the connectedness is, how many connected components are there? And we try to understand systems from that point of view. What I would like to do is to try to understand dynamics of these. Okay? When people do that, they usually come up with a model of the following type. Okay? So it's kind of one-to-one correspondence. I don't know how much you probably know differential equations. I should not be, right? So here are the unknowns, right, for each X, Y, Z things. There's some decay rates. These guys decay. If you don't do anything, they will just go down to 0. This is exponential decay. They just do this. And then for every one of these connections, so let me go to a Y1. Y1 depends on X. This is up regulation. This function over here looks like this. It's a hill function. This is not going to work very well. Goes like this. Monotone up. As X goes to infinity, this converges to 1. This alpha 2. If X goes to 0, it's equal to 0. So it's somehow a rising function monotonically up. And these come from biological, biochemical considerations. If you have a down regulation, you usually come with a function like this, which is a function which decays to 0. So the effect of Z on X, the more Z you have, the less X will rise. That's a canonical. Notice there's all these parameters. Okay? I can describe by a marker what they mean, but there is a 1, 2, 3, perhaps, per arrow, right? And then things which I did not put, but the decay rates, which are for each node, there's at least 1 power. These are unknown. Each of them costs about 3 months of a postdoc work, okay, if you want to get that. And some people don't get that. So the question is there something one can say about by dynamics how the system will behave without knowing this, or more precisely, knowing for all possible values of parameters? Okay, so here's a different model, okay, which is very much related to this one. I replace these ugly big functions by kind of these nice symbols which are a little more tight. But the key here is that those functions in the system look like this. So the Hill function model, which generally looks like this, are replaced by a piecewise constant function. Okay. But that does a lot of good to me, and I'm going to try to explain that a little bit. So when we engage in differential equations, why this could be a very important point. Okay. So how much you want to hear about differential equations? You want to hear more about differential equations? I'll just do more answers. Let me do a few pictures. Okay. Need to expand the horizon. So, very few pictures. I'll jump over things. Okay. So there now these functions. This function over here is right, so here's my network. X depends on Y and X. This is dependence on X. This is dependence on Y. Okay. How does this function look like? It looks like this. If X is less than 1, the function is 3. So, sorry, less than 1 is half, and bigger than 1 is 3. Okay? So it goes like this, goes uphill. The other function goes from 1 to half. So it's downhill, it's a down-regulated function. Okay? And the other one, this is also uphill function, going from half to 3. So if I look at the phase space, this is now where the X and Y behave. there are these thresholds. There's a threshold 1 and 2, 1 and 2, and threshold y. And so the behavior in each of these pieces will be different. Because if I evaluate my function right here, and evaluate this differential equation in this particular box, right? Well, I'm above 1, below 2, and above y. So these become all numbers, and this is what the differential equation is. Even if your differential equation happened a long time ago, you should be able to solve this kind of system. Okay? And in particular, solution is trivial, and if it's trivial, it means the computer can do it without your input. Okay? This 3/2 and 1/2 becomes, in the long run, the x and y approach that point 3/2, 1/2. Target point, and every solution here goes toward that point. Think about like a dog running after a big piece of meat. However, as you cross this domain, this threshold, things split. Okay? Because in here, suddenly your y went from above 1 to below 1, so one of these things changed to half or something. So every one of these has a different target point. Okay? And as you do this, you're chasing. So this is your target point, 3/2, 1/2 over here. If you're here, you're going to 3, 1/2. So you're going that way. If you're here, you go to 3, 3, which is here. You're here. So now you start to see that these differential equations can stitch these solutions together. And you basically get a very rough approximation of what the dynamics will be of these smooth functions. Each of these boxes, you can write down exactly what the solutions are. And instead of keeping track of all of this information in terms of solutions and individual pieces, I'm going to just look at the collection of these walls, which are these dividing lines. Okay? And perhaps, like in this case, all of these solutions converge to an equilibrium inside. So I have to put that object in, and I make a graph. So I combinatorialize the dynamics, make this into a graph. So we're getting closer to computer science, right? The normal differential equation. Right? So this wall over here is that dot. Okay? This wall over here is the green dot. This red is here, right? Now, all I'm going to say is that solutions which come here, going down, solutions here are generally moving towards that point, is this arrow. So just write these arrows down and characterize in a very broad sense how the solutions move around. And that's my resolution I'm going to keep. I'm not going to go and try to solve in any more detail differential equations. Instead of tracing individual analog of having fixed points and periodic orbits are now in this graph a strongly connected components of the graph. Okay, so look at this graph. Where is the strongly connected components? Is this one? I forgot to put an arrow here. Is this one? I capture those two and say, okay, I call them Morse set for reasons which are historical, but those are strongly connected components of the graph. Okay? So in my view, in the kind of bigger scheme of things, so this could be done in n dimensions. And by n, we could easily do this in 50, 30, or 50 dimensional space. Okay, so I have not, instead of 2 dimensions, I have 50 dimensions. And I can write this graph and tell you what the strongly connected components are. So take this dynamics and differential equations, and for those particular models with those piecewise constant nonlinearities, I can write each of these dots be one of those walls, put the arrows down, install it on the computer, and compute strongly connected solutions. Compute something which I'm going to say, this is a representation of dynamics. I have P3 would be, what do I see here? So this P1 is clear, this strongly connected component. P0 is over here. P2 is this one. And P3 is that one. Okay. So those are dynamic, so those are invariant sets, and flow between them goes the way the green arrows go. So for me, that is dynamics. More importantly, as I said, I would like to understand things across parameters. Pay attention for a few more minutes, then we'll go to malaria or something. Okay. So how does this dynamics, this graph, depend on the parameters? So instead of having these numbers, as I put up there, parameters will be B's and A's and actually the thresholds as well. Instead of having these functions, there you go. Look at this. So in this function, which is kind of a smooth function which people use, I have parameters alpha, x to the n, over kappa, and plus x to the n. Here's a parameter, here's a parameter. There's also not a parameter at the base. This is the base level of transcription, and this is the maximal of transcription, and whatever. These are corresponding very clearly to, if I make my function like this, the threshold is the lower value and the upper value. I call them B and A's over there. Beta is the lower value, U is the upper value, and the threshold is the kappa. So if you make this function steeper and steeper and steeper, with angle to infinity, we can approach that. There's a one-to-one correspondence between parameters in my system and your system. So let me now study how dynamics on a system like this behaves when I change A's and B's and thetas all possible ways I can possibly do. Here is the main observation. This is my dynamical system. This is my graph, which represents dynamics. Each point being a wall and how they map. Well, solutions in here move towards my red point. That's the target point. If I just move this point around here, nothing will change in knowing that this wall maps to that wall. Because no matter what it is, all these solutions will just kind of tilt, but they will always go there. So that map, this arrow, is going to be always there when the red is moving around. But if I move the red across all of these thresholds, suddenly things change, my wall graph changes. Now there's a new point. I have to introduce a new point, and now we can dynamics. So the only place where things change, well, there's one more place where things change, which I'll just go very briefly through. So there's a way to have the graph. If I have solutions go to here, and if I change my the wall graph, then move my target point, and the wall graph changes slightly. Okay? So my point is that I can completely understand. Sorry, I moved this from Keynote to PowerPoint. What I'm telling you that we know a priori, I can a priori compute a. So here's what. So if you think about this, there's discrete places where things change. So if I'm going to draw now a 100, this is a parameter space. I did not know that I can draw 100-dimensional space, but here it is. Okay. That's 100-dimensional space. That space, as I said, if I take some parameter, this is your AI, BI, your thresholds, many of them. If I just move them around a little bit, nothing changes. I can compute, so the space is going to be divided by these hyperplanes, which I can compute explicitly. Those correspond to target point right crossing the threshold. And each of these domains will have a constant wall graph. So this is called in dynamics a bifurcation diagram. We have to go and see here my dynamics changes from 3 equilibrium to 2 equilibrium. Those are important information. Here I can a priori compute this division into many domains. And I can represent each of those domains as a dot. And the neighboring domains which cross by an edge. So I can do something which is called a parameter graph. So the parameter graph is a division of a parameter space into closed domains where my dynamics is the same. I'll just finish with this. So my final description of dynamics is this graph of these yellow dots, which is called Morse common graph. It tells me the equilibria, periodic orbits, all these kind of pieces, parameterized by nodes in the parameter graph, which is over here. So, oops. Well, okay. So instead of this original dynamical description, which is called classical and dynamical system, we have parameter space, and then these things smoothly change. 3 equilibria. Sometimes you go out, there's only 1 equilibrium. Over here, the parameter space is divided into domains. Each domain is represented by a point. Domains in the parameter space are neighbors, if they are neighbors in that sense. And over each point, I have a description of what dynamics in terms of this cone Morse graph. Okay. That is for me dynamical system. So let me tell you how this works. This is just kind of showing you a pretty picture, but I'll get there. Here's my network, okay? And it has this kind of a loop of 3 things inside. If you know work of the Elowitz Liebler 2000, that's called a repressilator, which was constructed in E. coli with 3 repressing genes in the coli. It's awesome. There's additional kind of feedback over here, which is negative. And again, if you know the systems biology, if I have 2 negative feedbacks, that network should be able to do something like a switch. You should have a state where one thing is on, x1, the x2 is off, or x3 is on and x1 is off. So if I look at entire parameter space, what this network is capable of doing, right? There's no input to me except of that. Right? And I assign to each of them one of these functions, positive and negative, and say what are all possible values of, which I'm going to change all possible values of the values, all possible levels of threshold. So there's 1, 2, 3, 4 times 3, it's 12 plus 3, 15-dimensional space of parameters. And this is a complete description of how that parameter space looks like. So I'm going to try to explain what this color code here is. Okay? Each of those nodes represents the space in place in a 3-dimensional space where dynamics is something. So what is that something? So if you look at, say, red ones, these guys, okay? Those red guys are maybe 10. It is 10. I'm sitting in, this is 10. It's 10. This is 10. Okay. This looks silly to you, probably. This is just saying there's a fixed point. There's one and only one fixed point. This says there's two invariant sets. One is a fixed point. The one is something which is called full oscillation, full cycle. Things oscillate in the graph. And this is unstable and this is stable. And so these are all possible different dynamics a system can do. What you see, what are interesting pieces here, these parts are, there's 2 equilibria. So you can see there's a bistability. So all these parts correspond to bistable behavior. All of the ones which have FC in it correspond to cyclic behavior. This is produced in a microsecond on a computer, and you can look at and start to see what kind of dynamics you're interested in. In addition, each of these points comes with inequalities. If I click on this, it tells you where in the space what part of the space of parameters that represents this particular. So this is kind of complete rough information of how dynamics work. Okay. If I want to take the same graph and color differently, you will see a class of points which are all the light blue, which has a single fixed point, and I call it monostability. If I look at a class B, which is dark green, few points, bistability, C class green has periodic orbit, and there's only one point, which is something which has class, which looks like this. So it tells you about robustness of certain behavior in a phase space, in a parameter space. There's a ratio before these objects. You can find them. In particular, you can go and say, okay, I would like to get to 56. 56. Okay? You find those parameters. You can run your favorite smooth model because you don't trust these weird models, and that tells you there should be periodic orbit, and there's this periodic behavior on that of those 2 variables. So you can actually go back to the original models instead of blindly picking points and picking the dimensional space. And then for those points, picking initial conditions before dimensions, and run your solver, you know what dynamics is once before. So let me close this first part before I go to applications. The following observation. Okay. So here's how you would like to know if your system has certain dynamic behavior, bistability, periodic orbit, something else. You compute this database, and these could be quite large. We're now computing network with like 7 or 8 nodes, and that means the parameter is about 30 dimensions. And the size of the parameter graph, so how many of these domains you have to kind of represent as a point, you get about 125 gigs, right? Just to store that. And you get, a few billion, 10 billion points. So we're running kind of into space more than speed issues here. That is, so maybe I should address this. The database is an SQL database, so it's fully searchable by SQL language. So if I say search for desired dynamic signature, you go, I would like to find something which has 2 stable points. Boom, comes up, right? And then if I find this vast space and I found the image which I'm looking for, like this bistability, I can go then and refine using a more traditional models to find a match to my data. Okay. So here's what I'm working on right now. I was at Duke last week to talk about this. This is completely stuck in progress. We have no results to this point. Right? So we got some malaria. What's a malaria? Malaria is a disease, which is a very strange behavior. So if you do not understand the differential equation, at least you will understand malaria. So I'll talk about malaria for a second. So malaria lives in mosquitoes, liver, red blood cells. It affects this many people a year. A lot of people die. Huge fevers. How does it work? So in mosquitoes, it affects humans. I'm not talking about this much. That's where they reproduce sexually inside. These are not bacteria. These are protozoa. They're real small parasites. They come in the liver, multiply, release into human blood cells. This is what I'm going to talk about in a second. They affect human red blood cells. And then multiply inside. They emerge out in a very irregular interval. So Vivax and Placibron, which are the main killers, emerge every 48 hours, all of them. And so the immune system goes crazy, you get fevers of 102, 104, you can fry your brain, that's a big machine. Every 2 days, this kind of that. Nobody really knows why and how they do that. Because they infect 100,000 cells, and they live in 100,000 cells. So how come they know to come out every 48 hours? There's multiple explanations. They can have a very precise clocks inside, which clocks for 48 hours. That's not that easy to do, right? But that's possible. They can listen to your clocks. We have a circadian rhythm, which you put in a room to understand we have a circadian rhythm. They can understand the other signals. They can talk to each other. It's not very clear what happens. The biology is not very clear. So our goal is to try to figure out if there are, of the data, which I'll show you in a second. Our hypothesis is that if you can desynchronize this, it works. So if you get these guys to pop out within 6-hour interval all over the place, the immune system can handle this. Obviously, there's a big deal. This is why I don't have funny because it's not very clear. This is a high risk measure. Okay? This data has been collected in the Walter Reed Army Research Lab, and we got it 4 weeks ago. This is we don't. Nobody else saw this except you guys. This is genes, 5,400 genes in Clociprum. If you recognize, that's a lot of genes. Viruses have 9, 20 or something. This is big. This is like yeast. This is time in hours. And what you see here is a high expression in yellow and blue in low of the genome, basically the full genome. That means very periodically. This is high, low, high. The period is about 40 hours, 41 hours. Some genes come on here, and some genes come on later, but they're all periodic. This is remarkable on its own because this data is done in vitro in a dish. So there's no human to talk to, there's no circadian rhythm, there's nothing else. These guys are sitting on a dish in a freezer or whatever it could be. And the other thing which is remarkable is that, the falciparum lives in liver and red blood cells and mosquitoes. And so you would think that there'll be like a third of genome which is working in red blood cells, and a third of the genome is just quiet. It only comes on when you are mosquitoes because you do different things. And this it looks like 95% of the genome is also. So we don't know why. Okay? If you look at transcription factors, because our goal is to figure out who is making this rhythm, which are the genes, what are the genes which are making the rhythm happen. If I can understand that, we can disrupt that rhythm with harm. The transcription factors, which is only 450 of them, which are the putative, base oscillator. What happens next is there's a group which we work with in Duke, which goes into this data, and I will not tell you how they do it. There's many, many ways how you can approach this, but they try to detect periodicity in data. There's algorithms for that, smoothing the data, and then find the best fit. If I look at any pair of these genes, I'm asking, is this gene driven by the other one or not? Right? But every single pair can score their likelihood. So at the end of the day, you get this kind of a big matrix, 5,000 by 5,000, with the likelihood of which edges are which edges are there. So it's covered in there. Now, this is very suspect in some sense, because you're trying to figure out edges of network based on the shapes of the curves. And there's a lot of assumption going into how we do this. But we have done something. Over to the next, so this is done by my postdoc Green. You try to take this matrix and build some networks. Again, this is very much computer science, right? So and this is arbitrary. This will work. It will work at the end if you know that you find the right network. You take the top scoring edges, so which edges have the highest scores. You identify within that class those genes which have both something that goes in, which goes out. That's your VN. Then you have another threshold, which is how low scores you're going to admit. And you build basically a stronger connecting control. There's a way how you can build networks from this data, which is not too sophisticated. This is so for that. This is something that we can collect. So as you see, some networks are very small, and then as you go up, and you lower your threshold, you admit more edges, you get networks which are bigger and bigger components. So I talked about 4, 6, and 15, times allows, and we're trying to figure out those could be potentially networks which generate rhythm reciprocally. Here's a 4-edge network, positive, positive, negative, positive. That's a good news for those that know, because I have overall negative feedback, which should oscillate. There's a lot of mathematics which one can do here, and has been done very well. You can prove if there is a periodic order in the system, any system of this sort, then the maxima must occur in a certain order. So we'll check whether the model could generate the data which we got. And these models actually get you understand completely the information of how this works. And so the order of the model must be 188, which is green, blue, cyan, red, maximum. Green, blue, cyan, red. No, cyan is before that. That doesn't work. So we go and we can do this, and we say, well, this is not a network that generates that. This cannot be all to generate the data. So we can go and do bigger ones. For this, there's no theory. You can't do theory. So what we do, we run this database algorithm, right? We find 96 parameter nodes, which are these pieces in the parameter space, where I have a full cycle, where I know there's a periodic orbit. So again, I search through, again, this is a 30, how many nodes? 1, 2, 3, 4, 5, 6, 7, 8. 8 times 3 is 24 plus 6, 30-dimensional parameter space. And find all the places in the parameter space where the periodic orbit happens. We have another program which can take that and checks if the parameter, if what kind of maximum and minimum that periodic orbit can generate. And there is no match. So basically that, any one of those, none of those parameters will produce a periodic, they produce periodic orbit, but not the one which has this particular sequence of maximum. And if you are worried about, you should be worried about the following. Like for instance, so what is the order between red, yellow, and blue? Well, I have no idea. The data is very bad. Right? So we really put into the program and saying, look, the blue minimum, the yellow minimum, the red minimum, the green maximum, magenta maximum, cyan maximum, data, any permutations is compatible with data. However, that is followed by yellow maximum. That is followed by a red maximum. And then, you know, the blue max and all these minima over here, any permutation is okay. So you put that in your search and it says, is that something you can match with the model? And the model says no. It's unfortunate, maybe. 50-node network. And again, I can do analysis here completely. And basically, things don't work either. And this is hard to tell. You have to figure out, you have to pull things out. But the order of maxima, which is implied by the model, does not match this one. So what this allows to do is to test hypotheses, right? You would like to say, so are these networks, can we find networks which generate that rhythm? And these are not the right networks. So we have to search for them. So currently, what we're computing is a 40-node network, which is too big for our situation. So this would probably a couple trillion node parameter node. But we can do some creative simplification of these networks, which I will not go into. Instead of having this long chain of 5 or 6 nodes, you can collapse it to one node and make a little small networks. Again, these are still quite large because there's a lot of edges and nodes, but you can, again, you can discover the whole dynamics and test the parameters. So the computations are running. We're not there yet. We did not find any so far, no network which would be something which would be comfortable to talk to our colleagues and say, try this. We'd like to end up here. So this is our part. Right? This is hard. This is where the money comes in. And there's very, this is kind of shooting in the dark. We have collaborators in Thailand, which are the army, the Navy research person actually goes to the in the clinic up in the jungle 6 hours north of Bangkok, and we're trying to get data from people's blood, because there you will see genes of the falciparum and also gene of the uterus, especially as a human, because we all like to know how these guys work. And as you may or may not know, that's hard because these people come with sick with malaria, and you say, oh, I would like to take your blood every 2 hours for the next 4 days. How about that? So the institutional review boards are not very happy about it. So we have to be very careful how much blood you can take. You have to give them medications immediately. You can't just keep them on the whole life to make an experiment in which you suffer 2 bouts of things that you need. So that's in process. We don't have that yet. I think I should finish here. There's other things which one can talk about. We're trying to think about what is the natural rhythm, what is the driven rhythm. There's actually some genes which are only 20-hour, maybe 20-hour period, not 40-hour period. So the question is, should be concentrated in a 20-hour period, which is smaller set than 40. What is easier to train? Probably 20-hour period, which is possible goes like this, and then have 24 hours to get a driving in. Then in training is easier than if you do 2-to-1 training or 40-hour. Anyway, there's a lot of stuff which you want to think about. And I'm extremely excited that this is, as a mathematician, I can make a little bit maybe contribute to this. Hopefully it will work. So let me just close here. So this, in this project, we wrote a self-contained toolbox, right, which goes from, which can produce these experimental hypotheses, starting from a high throughput data expression like this. And I would like to say, here's a network which produces this data. Whether that's feasible, we'll see. So with this thing, malaria, this can be done on cancer cells, microbiome, and many different sets of data. For the tool development, this database which tells you about dynamics of a system across large parameter spaces. Currently, the storage space is worse than computation. We think we know how to address it a little bit to store less data than needed. And then the big thing which we got to do is these pieces of software as always are, this is a breaking Python here, this is a vectors and that, and this is, so we have to make it into a nice atlas when we do some of production level code rather than kind of for each thing. So that's, I think, all I want to say. And I apologize one more time. I was 6 weeks ago. Thank you very much.
So we can take 2 questions and then we'll break.
So at the metabolic level, a lot of these interactions between products with these genes are going to be more than just pairwise interactions.
Yes.
There's a lot of enzymes binding point reactions.
And there were some, but there's some of the methods we had. It was not just a simple loops. There was some interaction between two ways.
Okay.
Yeah. So that is not right. Yeah.
So all of the nodes in these regulatory networks, though, each node corresponds to a single gene.
Yes. Genes or metabolic something.
Okay, so you can incorporate metabolic information. It's not just gene expression.
So here's a challenge. Right? What I'm telling you is that I'm telling you the interaction between them is well approximated by a stepwise function. So I'm saying my effectiveness is low, and some people high. It doesn't have to be stepwise, but it should be increasing. That happens quite a bit in gene regulation, where you have multiple enzyme reactions. Where that's happening in biochemical reactions, where we really have both of the constituents well represented, that's not good. So I think I can, right? There's nothing preventing me that, but I have to, all these interactions, coding should be fast. Now, that's a computation model. Once I found the dynamics I want, I can go back and try to smooth it up and see that things are found persistence. So the principle is
Any other questions?
If no one else does, I have a follow-up. Differential equations response time ago. Am I correct that the fact that the boundaries between domains when they're constructed with homographs, the fact that they're orthogonal comes from the using that switching model?
yes.
Okay.
Yes. Yeah, looking for was one, yeah, absolutely. Because I have a threshold, right? The threshold is X and Y. So that's what I follow. Okay. But the dynamics of it does not. 5 o'clock Monday, 9 seconds.
Looks like he wants to get.